So we've got another session chair for you this afternoon who you've not met yet, and I'm going to introduce him now. So this is Professor Gary Black, and he is another co-director of the Hub for Biotechnology in the Built Environment. So we're really delighted that he could join us today. Gary is Professor of Protein Biochemistry at Northumbria University, specialising in the discovery, development, production and analysis of biocatalysis enzymes and their applications for production of high-value natural products. Gary is currently co-director of the HBBE, institutional director for the BsIA, which is the Bioscience for Sustainable Consumer Products, BBSRC Collaborative Training Partnership, and co-lead of Northumbria University's BioFutures Interdisciplinary Research Theme. Most of Gary's research involves collaboration with industry, and in particular he's currently working closely with Procter & Gamble as part of this BsIA CTP and associated projects. So thank you very much, Gary.
Okay, so this is session 2, Engineering Biology for Future Textiles. So we have 3 speakers. We have Roberta Morrow, who we have here, Anastasiya Kishkevich, and Katie Gilmour. So just a little bit about Roberta first before we start. So Roberta's research targets the demand for sustainable material innovation by exploring design, biology, and textile manufacturing. Her approach to new manufacturing routes for biotextiles focuses on utilizing craft knowledge and material exploration. By doing so, core understanding of material characteristics can be explored to design sustainable textile manufacturing. So Roberta is currently a postdoctoral researcher at the Royal College of Art, and she works closely with York University. Okay, Roberta.
Hi, hello, can everyone hear me? Is that all good? Okay, great. First of all, thank you so much for having me in this space in the ARCINTEX. Thank you so much to Jane and Romy for having me here. A little bit of context on my background. I was a structural textile designer, and then I moved into material circularity and textile manufacturing. I then moved into biology as well, and I worked here at the HBBE for a time with Jane, looking at biomaterial manufacturing. I'm now a postdoctoral researcher on a couple of projects within the RCA, one of them being the Textile Circularity Centre, where we work with multidisciplinary universities, including Cranfield, Manchester and York, and the Biomanufacturing Textiles from Waste project, where we are in combination with University of Leeds and the University of York. So today I'll be talking about the BBRSC Super Follow-on Fund, the Biomanufacturing Textiles from Waste, where we have an amazing collaboration with York Biology, where I'm a guest research associate in that space, alongside the Royal College of Art and the University of Leeds. And within this, I'll be talking about what my role within the project is, which is where I look at turning biomaterials into fabrics, yarns, and textiles. So a little bit of context on the material industry at the moment. I'm sure we all are very aware of the problems of the textile industry. But the main ones that the BBRSC funded projects are looking at, or this one in particular, is the idea of raw material consumption and the end-of-life waste streams. So as we make new materials, we are harvesting lots more raw material consumption. It goes through productions, transit, transport, and then has various recycled life cycles, whether it's through take-back schemes, recycling, donations, use phases. And then ultimately, it will end up in landfill or incineration, mostly incineration, as we found from our research. And so the key areas that we are looking at, as I said, is looking at the idea of reducing the land mass that we are consuming of raw materials, and also the idea of using this waste stream as a fermentation process in a biological sense of creating new materials. So we're looking at using the waste life to avoid over-consuming of land consumption. And as you can see at the bottom, we must utilize materials already in circulation. Oh, I think the clicker is gone. Okay, so I won't go into too much detail on this, but this is our process, and the amazing team at York University, the biology team, have created this bioprocessing way of turning end-of-life waste. Within this project, we use wheat straw into using a bioprocessing to create new materials such as bacterial cellulose.
Sorry,
Should I say what has fun? Cool. Okay. Yeah. So the people on the live stream, we are looking at using a waste material in a bioprocess to make bacterial cellulose. And here is our beautiful sheets of bacterial cellulose that York had created with their bioprocessing. And where I come into this research project is that we're looking at how we can utilize these biomaterials and turn them into pliable textiles. In my area is structured knitting, so I look at how I can turn them into fibres, yarns, and etc. And I think at this point, the project, I kind of turned it on its head a little bit, because I think what the idea was that I was going to go through traditional textile streams, where I would take the bacterial cellulose and use a very traditional way of creating fibres through regenerating cellulose, which through wet spinning. And I have done this in the past, and we are in a separate project, well, same project, but in a separate area to me. Leads are looking at wet spinning bacterial cellulose. But for me, the question was, why are we taking this material that's growing itself? Ooh,
So
yeah,
sorry
no,
to
of course.
interrupt again. We just got feedback that the slides are not sharing online. So we try to stop sharing and sharing. PowerPoint. And we'll give it another go.
Yeah, just let me know if it's
Yeah,
Yeah,
just
all
saw
good?
the slide.
Okay, so sorry, yeah, no, so I had a trouble with the idea that we've got these new materials, well, not so new, but designing these new materials, and we're putting them into established streams that aren't necessarily meant for them. So instead, I kind of went to my supervisors at the time and said, look, I really want to look at the material characteristics of these and see how we can design with the biology, instead of putting it into traditional streams, to maximize the sustainability of the production system. And I suppose going back a little bit and just to have a look at what's going on in the industry, I'm sure you've seen many examples of these amazing designers, ranging from packaging to fashion, apparel, to indoor architecture, to all the way to new modern synthesis using new material systems with new material biologies. So I guess these are my kind of three really key questions that I want to ask within my own research is, how do we make sure that these new materials that we've created from biomaterials, there's lots of, potential greenwashing going on and buzzwords, how do we make sure that they are actually produced in a sustainable way? Because a lot of the time we get these new materials and we put them into a system that's already established, and those systems might actually not be the most sustainable route in terms of life cycle assessment. How can we utilize and enhance the inherent characteristics of new materials to create the most sustainable production of the yarn and fibres, designing new material systems and manufacturing routes around these characteristics? And one that I didn't put on the slide, but it's very core to my research, is that I want to make these, or try to make the community, as we all do, accessible to everyone. So it's not that we're making these new materials that are high-end designed, they can span from that all the way across into different communities. So my principle of designing, and I know that a lot of us will be using this kind of structure in our own work, maybe potentially without even realizing, is that this key design knowledge, biological understanding, and knowledge of textile manufacturing scale, and how they can all work together to create really informed manufacturing textiles of new biomaterials. And this can, again, be multidisciplinary, multi-collaboration, as I know the HBBE are very, fond of, and also this idea of exchange of knowledge. It doesn't all have to be within one person. So going into a little bit of depth, what is design knowledge to you? And to me, it was very much installed very early on in my, academic journey, with Jane being my very much a big mentor in my life, that material design and design knowledge really comes from the way that we interact with materials. And we do it anyway. We go into a shop, we touch material. How does that feel? What are the characteristics? And the idea is that you're getting curious of these new materials from a very almost naive perspective, but not naive at all, because it's the inherent craft design knowledge that we all can carry if we know how to extract it and intuitively apply it. But then what we can do with that design knowledge is start to get really informed backgrounds of the biological and what is going on within these growths. How can we edit the characteristics for future textiles? So by interacting with the materials, you've got that design application, you've got that design knowledge, and you're curious, you want to find out more. This is definitely what happened to me in my journey. So actually having my residency, having my guest associate role within York has allowed me to really understand the biological environment and growth of bacterial cellulose. Yeah, so what are the characteristics, what are the biological characteristics, how can we use it, and how can you as a designer be implemented into the biological stage instead of after? Because a lot of what I've found is that as designers, you're kind of given a material and say, go make something with this new material, but why are we not being implemented right at the beginning where we can start to make a difference? And then the third thing is the idea of textile manufacturing stage. And originally this wasn't a part of my, this is the idea, but as I moved through my research, I realized that actually the knowledge of fibers and the knowledge of textile manufacturing really implemented how I saw the outcome of these projects developing. So the scale, how can we scale? How can we start to think about digital technologies? What are the characteristics of those materials that are going to make it manageable for a society that demands new materials? So I guess I'll go into a little bit of the case study of self-constructing bacterial cellulose fibers for application in structured textiles using this kind of methodology. So from these slides are a lot of trial and error and a lot of failed experiments. But on the left, we see kind of the first ideas of craft applications of the material, really getting involved with applying it into craft processes and how we can then develop it. In the middle, I really spent a lot of time with biologists and learned so much about the way the bacterial cellulose grows and the way that the environments change and how we can then adapt it to change those environments. And it's a bit of a running joke in New York because I'm the crazy designer that comes into the biology lab and says, oh, let's try this, let's try that. And everyone goes, what are you doing? And everyone says, oh, you're making some more spaghetti. That's our running joke instead of making yarns and fibers. But in this process, there was a lot of trial and error fails, which is where I learned the most about the material. And finally, through a lot of trial and error and, convincing people that I was on the right track, I managed to derive a way that we can change the environment and change the physical qualities of bacterial cellulose so it starts to spin itself into a fiber on its own. And the sustainable idea, this is a picture of me when it's the very first time it worked. You can't see my face, it's a big beam smile. And the very first time that it worked, it was great, and I'm really happy with how it turned out. There's so much more that I can now do with it. Sorry, I've lost my train of thought now. Where was I going with this? Self-constructing bacterial cellulose fibers for application in structured textiles. And yeah, so the idea is that there was a lot of trial and error, and we can edit the environments depending on what you want and what your characteristics want and what the characteristics of the fibers need for the application. So this is where my outcome came to of self-constructed fibers. And the most sustainable thing, or in my eyes, the most sustainable thing about this is that we are growing it directly from the growth of bacterial cellulose. So you don't have to then put it back into a stream of dissolution where we're taking the DP down into a dope, spinning it directly out into a fiber. Because I think a lot of the new materials that we're seeing in the market, especially the ones that have the qualities that we desire, the processing is the same as potentially viscose and lyocell, which we know have questionable ethics in terms of production. So with this process, we can directly spin the fibers and yarns from bacterial cellulose growth. And that has come through the methodology of learning about the material designing and textile manufacturing. Ooh. And yeah, and there's lots of things that I am hoping to do with this. There are still ways that I want to tweak the growth. We're changing the scale of going from macro to micro, quite well actually, going from how I can spin a cellulose that mimics more of a cotton, then we can then translate into more of a fibrous short yarn. How can I think about editing the growth so that we can start applying it into digital textiles? And most importantly, how can I, make sure that this is an accessible route for scalability, but also different communities, so that biomaterials can be accessible to a lot of people. And I'd just like to give an acknowledgement to the late Professor Simon McQueen, who was a massive influence at the beginning of this project. Thank you.
Questions?
Thank you a lot for the presentation. It's a sort of viscose process, no?
In terms of what I do here,
Normally the viscose is a sort of cellulose too, but we use some chemical for that. I don't remember which acid is, but it's not really good. How you fix this problem, like how you make chemically the system for make a filament with sustainable, because normally this system for make filament is a sort of different batch with a sort of liquid, or we can make this by drying too, but it's not really good. So most of the time for make filament, we need to use some acid. So how you fix that with a sort of green chemistry? I don't know, just a question.
yeah. I mean, so that was the whole idea, is that I wanted to cut out this phase. To me, we take a cellulose-based material and we obviously, like you say, we put it into dissolution through chemical, and then we wet spin it into a new fiber. And that for me is adding in a step that we don't need to, if we know how to then edit the environment of this growth. So that was the principle of what I was trying to work towards, is the idea that we don't, just because we have an established system, doesn't mean that we have to put these biomaterials into that established system. In fact, I absolutely encourage anyone to completely think outside of the box when it comes to new materials and really question themselves on, is this material actually sustainable? Is it going to be coming from a sustainable source? Does that answer your question? Sorry. Yeah. I found it that way.
I think to kind of jump onto his question, from what I'm understanding, what he's trying to ask is was it a sort of gene editing that you were doing, or and then are you creating like an environment for the cellulose to specifically grow in a filament shape? Like, is it growing in a mold, or like is it just like in a vat and it's like floating in liquid and then it is naturally spinning itself into a filament? Like, how is this process actually happening?
Yeah, okay, so basically the way that I understood of bacterial cellulose growth is that it wants to get to the nutrients, it wants to get to a nutrient source, and it wants to get to the oxygen. So I really utilize the idea of guiding the nutrients, and so the bacteria will then follow and secrete the cellulose in that shape form. And to do that, there was definitely multiple different things that we can try. I think the next step is definitely genetically engineering bacterial cellulose. I want to be able to try and organize the fiber, the way that the filaments grow together to create the biofilm. I want to be able to organize that so that we can make them more aligned. And I think that's where the textile knowledge really comes in as well, the manufacturing knowledge, because you can then have a look at fiber science. How can we then start to implement fiber science into the biological growth? It's all of those kind of questions. But yes, to answer your question, it is definitely a combination of physical growth and biological editing. And the next stage is going to be, how can we make the microfibrils, the ribbons, softer, and how can we make it less paper-like, and how can we make it more lustrous, more textile-like, by doing that chemical editing.
So, congratulations for this amazing feature. I was very curious to know whether have you tested the hydration capacity of this cellulose material, because being a bacterial cellulose, it's known to have like about 90% more than sweet, the water capacity of holding it.
Yeah.
So how do the weave patterns work on adding water? Do they untangle themselves, or what happens here? I'm just curious.
Yeah, no. And so as I say, this material is directly grown from the bacterial cellulose, so that is completely the same. It has major capacity to hold water. And a lot of my research before this was, especially in my master's, was again questioning the idea of why are we trying to change the inherent characteristics of a material. So a lot of my research was to do with wound dressings and the idea of absorbing moisture, which is where I started looking at bacterial cellulose, because in my mind, instead of adding in something to make it hydrophobic, why don't we utilize the idea that it's hydrophilic and hygroscopic. So yeah, it's a really interesting avenue, and it's very important that we look at the idea of making it waterproof. But in my mind, I want to be able to really understand the characteristics of the material so that we can apply it to the right materials instead of just applying it to everything. Yeah, but yes, it is. Sorry, that was a short answer. Yes, it does exactly the same as bacterial cellulose, so it does absorb a lot of water.
Okay, we probably should leave it there.
Cool, yeah.
So let's start with one more time.
Thank you.
So our next speaker is Anastasiya Kishkevich. Hope I'm correct in pronunciation there. From Imperial College London. So during her PhD at University College London and first postdoc at the University of Oxford, Anastasiya was focusing on basic research in yeast as a model organism. She joined Tom Elser's group in Imperial College London to work in synthetic and engineering biology and to use yeast in production of engineered living materials.
Yeah, okay. Thank you very much for introduction and for giving me the possibility to talk to you about how kombucha co-culture has inspired us to generate this engineered living material. So kombucha is fermented drink made from tea via symbiotic culture of bacteria and yeast, or SCOBY. And the main bacterial component of kombucha is Kombucha species. And yeast part will be represented by various species, including Saccharomyces cerevisiae, which is just usual baker's yeast we use for bread, beer, and all the different stuff. Okay, so, and this, you can see, this is what you don't see in the supermarket. This is how kombucha is actually made. So bacteria is fermenting tea, producing acetic acid, and also producing this pellicles, we call them pellicles, bacterial cellulose, what Roberta was talking about, but this is how it is made in traditional way, basically. So just to recap this marvelous properties of bacterial cellulose, it has high purity, high crystallinity, high water retention, good mechanical properties, forms 3D nanofiber structures, is biocompatible and even eatable. So these are all the advantages about the usual sourced plant-based cellulose, basically. And as Roberta already mentioned, it's been used already in design, cosmetics, medicine, and other applications. So what we thought, okay, can we do a SCOBY culture in the lab? So just to mention that most of the work I'm presenting today was done by Dr. Charlie Gilbert and by DJ in collaboration with Team Lab in MIT. So DJ was the PhD student at the time there. Okay, so what Charlie did, he decided to grow, as I said, kombucha culture in the lab. So here we have glucose as a source of carbon, and it's also a source for cellulose production, because this bacteria uses glucose to build up cellulose fibers. And the bacteria strain we are using is K. reticus, which is actually was isolated from kombucha during IGM project. And the yeast, as I said, this is just well-studied, well-characterized laboratorial strain of baker's yeast. And you can see that in glucose, bacteria can form nice pellicle by itself. Obviously, there is no pellicle for yeast, and the co-culture also forms a pellicle. But the problem here is, okay, so bacteria actually can form the pellicle, so she doesn't need yeast there. And it appears that bacteria in these conditions outcompetes yeast, so it grows much faster, uses all the glucose, yeast doesn't have any, so these don't really grow. So the idea was to substitute glucose to sucrose. because bacteria can't grow in sucrose, while yeast secreting an invertase, an enzyme which can break glucose into sucrose and glucose and fructose. And then these monosaccharides will be used already for bacteria to grow cellulose. So now you can appreciate here. So when we have sucrose media, bacteria come from the pellicle, while we can see the pellicle in co-culture. So after the test and trial attempts, Charlie has identified this unique combination of how much cells you need to put of yeast, how much bacteria you need to put, so that you can get actual co-culture growing and yeast being incorporated. And here you can see this cool picture of 3D cellulose with yeast incorporated, and it was done by all. So we see, yes, yeast can incorporate it. So let's functionalize our bacteria, but about bacterial cellulose, and how we can do it by modifying yeast here. So what we can do is to genetically modify yeast to give us properties that we want, and then yeast will be this sensing and output component of cellulose, and yeast will be incorporated in cellulose. So cellulose will have certain properties. Okay, so an example here I want to show you is the beta-lactamase enzyme. You see that enzyme will be secreted by yeast and is incorporated into cellulose. And what beta-lactamase does, there is a reagent nitrocephin. You apply the nitrocephin to bacterial cellulose, and if beta-lactamase is present, that the pellicle will turn red. And this is basically what Charlie has observed here. So we see the 3 sets of pellicles. One is just wild-type yeast, then the one that has beta-lactamase, and the third one is the beta-lactamase with CBD, which is cellulose binding domain. So meaning that the enzymes will be actually solidly attached to the bacterial cellulose. And you can see that the color develops quite quickly, and this more color developing for beta-lactamase with cellulose binding domain. But what is more interesting is that we can actually drive cellulose, and then rehydrate it, and the enzymatic properties are still there. So it can be stored for a while. You can still see that enzyme is active. So with certain storage life for this product. Okay, so this is about the enzyme, but what about yeast? So what we can do with yeast sensing? So Charlie went to develop this living biosensor made with bacterial cellulose. So what we have here is the yeast, which has the green fluorescent protein integrated into it. And when we add beta-estradiol or BET, you see that the green fluorescent protein is turned on. So yeast will be red, while they are not red before. So here again, this is a grown pellicle, and then you can dry it. And if you incubate it with inducer or without inducer in the media, letting yeast to grow and propagate, you can see that indeed there is green cells present. And I apologize for somebody if somebody can't see green. So you can see that the pellicle dried, and in 3 conditions, we still have yeast there present. And even after 4 months storage, you still can see yeast recovering and living. So showing, okay, the material can actually sense output and signal. So this was a proof of concept. And what we were thinking, okay, how we can modify properties of bacterial cellulose, make it responsible to environmental stimuli, such as heat or water, pH difference. And what we thought about is to incorporate elastin-like polypeptides into bacterial cellulose. So elastin-like polypeptides are these multiple repeats of valine, proline, and glycine amino acids. This is highly repetitive, not really like to be produced really and secreted. But the idea is that we combine yeast secreting ELPs, and you can see that they are very elastic here. Is like if you change pH from liquid, they turn to fibers. So yeah, we combine bacteria with producing cellulose with ELPs, which will also have cellulose binding domain. So they will form networks within the bacterial cellulose, and this will change, hopefully, even elasticity of the material. So then there will be physical properties changes, but also response to the environment. So, and how we can achieve it is we need first to engineer yeast, which will secrete ELP into the environment, and also optimize co-culture so that we have as many yeast as possible, producing as much protein as we can. So to generate the yeast, what we need to consider is first promoter. So this is a part of gene which regulates how much of the protein is produced and how much gene is expressed. and also the secretion signal, which will regulate and guide the protein guide the protein outside yeast cells. So we don't need to harvest yeast, we will just let them grow and they will naturally secrete it. But unfortunately, there is no unique combination of these components for any protein. So you really need to test different ones. And to avoid this testing one by one, what I'm doing, I'm combining the library of promoters and secretion signals together. And via this approach by surface display, basically, so we have all the parts of our protein being attached to the surface of yeast when they're secreted. And then there is another tag which will be recognized by, for instance, antibodies. And we can analyze it with the fancy flow machine, and then to detect how much of this fluorescent signal we get as an output. And you can see here this yellow, bright yellow peak. This is the maximum one we can get. So all of this, we don't have any signal, while this one is the combination of promoter and secretion signal, which actually gives us high yield of expression. So using this approach, I'm identifying this genetic modules combination for 10 different ELP repeats. So I have ELP 10 repeats of this valine, proline, glycine amino acids, and then this is multiplied by 10 times. So that makes them very not being likely to secrete it. So they're very repetitive and structured. That's why it takes quite some time. But basically, at least for now, I have the combinations for most of the ELPs I'm working with. And the further steps to take to generate our environmental responsible material is to increase yeast incorporation into cellulose. And what is happening in the kombucha culture and the same in our cultures is that yeast bacteria, being very acidic bacteria, will produce acids. And if we start with the pH around 6.87 in the media, very quickly it will drop to 3 to 4 pH. And at 6-7 pH, yeast are very happy, and as bacteria as well. But then when the pH lowers, yeast becomes sad. They don't really like anything below 4. And we ended up here with yeast not surviving there, so having very low incorporation into our pellicle. So the way to approach it is we're engineering yeast to tolerate lower pH levels, and also trying different buffering medias to increase the incorporation of yeast. But obviously the more radical one will be to use different yeast, which we don't really want, because Saccharomyces cerevisiae has this very nice genetic toolkit we can use. And the final thing will be also to increase secretion of ELPs, because obviously there is a limit, and we can engineer yeast introducing mutations or allowing some genes being expressed more to get more ELPs as much as possible into our pellicle. Yes, and that was very fast. So yeah, this I would like to thank everyone for attention, and then thank Tom. He will talk about bacterial cellulose more, and the whole group. And as I said, most of the group, the proof of concept was done by Charlie Gilbert and CJ Tan. And yeah, this is it. Thank you.
Nice talk. Just on that last slide regarding pH, have you guys thought about generating a cellulose producing bacteria that knocks out the acid producing genes, just to try to combat the pH issues?
Yeah, that would be very interesting. But first of all, this is a bit trickier to engineer Curie because there's not too much tools. And secondly, it doesn't really like to be interfered with anything. Like we tried some other modifications, and it really affects cellulose production. So it doesn't even like being the buffer in media. So if we do artificially lower pH, and like, yeah, I don't want to form pellicle anymore. I'm out here.
Thanks, Anastasiya, for a nice talk. You may have said this, I mean, this sort of wide engineering of the yeast, and is the ultimate aim to engineer both the bacteria and the yeast to have the most efficient process of bacterial cellulose production going forward?
Yeah, so bacteria, thank you for question. So bacteria, if there are other bacterial species and strains we can use to enhance cellulose production, so there's no really need to make more cellulose. So we're using it as a, not scaffold, but at some extent a scaffold for yeast being there. And as far as I know, people who are trying to increase cellulose production, they end up with like 10% increase, which is not that's something that you really want to invest to, I would think, too much. But yeah, for now, yeast is easier to engineer. We engineer yeast, but there are other people in the group who are looking into bacteria engineering.
Questions? We could give Anastasiya a round of applause. Thank you very much. Okay, so our final speaker of this session is Katie Gilmour. So Katie's from the HBBE, a collaboration between Newcastle University and Northumbria University. Katie's interested in sustainable research with interdisciplinary themes around microbiology. She recently completed her PhD at Newcastle University, which was a collaboration between geomicrobiology and geotechnical engineering. She investigated the presence, activity and survivability of microbes in clay intended for use in nuclear waste repositions. Currently, she is working as a senior research assistant on a range of projects within the HBBE at Northumbria University, including the bacterial cellulose project. Over to you, Katie.
Brilliant. So, yep, I'm Katie, and as Gary said, I'm part of the HBBE on the Northumbria University side. I'm a biologist by training, and today I'm going to be sticking with the bacterial cellulose theme and talking about how we used an engineered biology approach to functionalize it. I'm going to be keeping it in reference to textile application. So as we kind of heard from the last two speakers, bacterial cellulose tends to grow as thin sheets and liquid air interfaces. And considering textile, it does have some advantages over plant cellulose in that it has a higher yield and it is of a higher crystallinity and purity, which just means that it needs less processing before downstream treatments can occur, like if you were to dye it, something like that. And in terms of textiles, those thin sheets, or the kind of pellicle that forms on the top of the liquid, most closely resembles a non-woven material. We've got researchers such as Suzanne Lee, who use the kombucha method of producing bacterial cellulose and then use those sheets to create garments. We've also got companies such as Nippon Paper that have created a bacterial cellulose paper, and an Australian company called Nanolose, which actually uses it more like a yarn, it's more like a Roberta thing, and then can create apparel that we're maybe more used to seeing in shops. There is one downside to using bacterial cellulose as a textile, or one of a few, in that it's highly hydrophilic, so it loves water. So if you were to put it into an environment where, or a microenvironment where that water content might change, such as between your skin and your clothes, if you were to get a bit sweaty, it would then take on that moisture, morph out of shape, and just be a little bit less suitable as a textile. Currently, industry does have some ways of getting around that, but they're not the most sustainable. So the first one uses a lot of high energy, involves playing sound waves through the bacterial cellulose as it grows. But a lot of the other ways are actually post-production. So after that bacterial cellulose pellicle has been made and harvested, you can apply chemicals to it, which tend to be quite toxic. So they've got quite a high environmental cost, as well as being quite a big cost for industry to have to deal with those waste streams. And another way is to actually use conventional fabrics and fibers and then use the cellulose as a coating to take on some of the properties of those fibers. But obviously we want to create a new engineered living material and preferably not have to rely on the older ones. So what we did was we looked in nature to see how naturally non-wettable surfaces or barriers occur. Mainly thinking about the bacterial world, And we found a protein called BsIA that comes from Bacillus subtilis. It is involved in biofilm formation, and it kind of creates kind of an elastic skin over the bacterial population, almost like a raincoat for it. So it sounds like something that could work pretty well with bacterial cellulose. We took that and we synthesized it with a cellulose binding module, just to make sure it's stuck onto that cellulose. We put it into an IPTG-inducible plasmid, overexpressed it in E. coli, and then harvested it to use it as a cell-free extract. And we did do a few experiments to see if it would be better for use as a purified protein, but actually in terms of the hydrophobicity, it performed significantly better when it was in the cell-free extract. And there was something in the literature to kind of explain that, in that when it's purified, it kind of clumps together, it aggregates a bit more, and it just doesn't perform quite as well. So I think there's something in that cell-free extract that just helps stabilize it. Now this image here, I'll do it on the screen for those on the live stream, is the predicted structure of our protein. So this blue area at the top is the hydrophobic head. Then this pink and gray barrel region is the rest of that hydrophobic protein, the rest of the BsIA. Then got this big loop, which is the linker region, which AlphaFold didn't quite manage to predict the structure of perfectly. And we've then got these two red and pink regions. That's our two CBMs. We've got a double CBM on this protein. And for our application, what was really important was the orientation of this protein when it sits on the cellulose. So we really want this hydrophobic head to be pointing outwards, so pointing to where the water is, so that we're definitely repelling it. And obviously, because we don't know the exact structure of this linker region, we can't say for sure what orientation it will be when it is on the cellulose, but I'll come back to how we think we can get around that and improve that later on. Oh. Okay. So after we'd made our protein, we had two things to check. The first thing to check was that it was in fact hydrophobic, and to do that, we just applied our cell-free extract to some filter paper, either in a ring around the edge or in the center, and then flooded that filter paper with colored water. The idea being, if it is hydrophobic, the colored water won't be able to penetrate the regions where the protein is. And happily, that was the case. The second thing we had to check was if the CBMs did actually bind cellulose. And to do that, we actually used a green fluorescent protein in place of the BsIA, just so we could see where that protein was. And we used that cell-free extract, grew some pellicles, and let those pellicles and the cell-free extract shake together for an hour, then washed the cellulose afterwards. And what you can see is that when we've got our GFP with the CBM, it's super green, that the protein's really stuck on there. Seems like it's working. And just to check that, we had a control with parafilm, no protein stuck onto that, so it's definitely cellulose specific. And then just to make sure it was definitely cellulose binding module and not the GFP, we have another control, which is cellulose with the GFP without the binding domain. And there's tiny little flecks of green on that, but certainly GFP with the CBM is where all the protein is. So actually then applying it to bacterial cellulose, we picked a couple of different ways to do this. The first one is ex situ, so it's more of a kind of coating method, more like what industry is kind of doing with a lot of their treatments at the moment in their post-production functionalization. So this was our cellulose was grown, we harvested those pellicles, dried them, applied our cell-free extract, then washed them again. Our other option was an in situ approach, where we actually added the cell-free extract to the growth media of our bacterial cellulose producing bacteria. And I'll just explain what's all on this plate. So everything's in triplicate. At the bottom, we've got our negative control on this side, just got off the screen here, which is just how the bacterial cellulose would go normally if we weren't doing any interventions. Then we've got an empty vector. That's everything that's in the cell-free extract apart from the hydrophobic protein. And then we've got our hydrophobic protein with the cellulose binding module and a hydrophobic protein without the cellulose binding module. And there are 3 different concentrations. C is low, B is mid, A is the higher concentration. And what we found that was really interesting was that the more hydrophobic protein we added, the longer the delay was for that surface pellicle to form. And I think the reason for that is that it's a hydrophobic protein. It doesn't want to be the liquid. It wants to be at that liquid air interface. So it's actually taking the position of where the bacterial cellulose wants to be. And actually what's happening is the cellulose is still being produced. It's just under the surface. And until it starts incorporating that protein into its network, it can make that space for itself on the surface. So as we left this for longer, we did start to see surface pellicles forming. The big question was, now that we've applied it, did we actually succeed in making the bacterial cellulose hydrophobic? And for the ex situ, that coating method, the answer is yes. So hydrophobicity is defined as being a material that has a water contact angle of over 90 degrees. And what you're seeing here is the best water contact angles we got for each treatment. So you can see A and B, so BsIA with and without a CBM, it's well over 90 degrees. With the in situ, it's not so good. Now, the reason we think that is that both treatments had the same concentration of protein, whereas with the coating experiment, all of that protein is on the surface. In the in situ, it's integrated throughout all the layers of the cell, of the cellulose. There's maybe only a small proportion of it that's actually on the surface. And it's still significantly better with it than without it. The only thing to note here is that in the controls, they're a bit lower on the in situ experiment, and that's just because the pellicles weren't growing for as long. So we wanted to catch them just as they formed, as that was the point that we saw the protein had just been incorporated fully into the pellicle. So to check to see if the protein was there, and if we were kind of right in our thinking of why those hydrophobicity results were as they were, we did some AFM imaging. And again, I'll start with the coating. So that's the top row on this slide. BsIA with the CBM, so the hydrophobic protein is completely covering the cellulose. You can't even see cellulose fibers in that image, it is just a sea of protein. The hydrophobic protein without the cellulose binding module, there's some clumps of it, but you can see the cellulose. It's definitely not as well stuck on as with the cellulose binding module. And then the active vector is just cellulose, there's no protein there. With the in situ, it's a bit of a different story. So you can see that it's really evenly integrated in the BsIA with the CBM, and it's also integrated within the layers. It's not just sitting on the surface, and it seems like quite an even coverage as well. The hydrophobic protein without the cellulose binding domain is just on the surface. It's not being integrated into it. I think that's just purely due to the fact that that cellulose binding module allowed it to be pulled into that matrix a bit more. The anti-vector image I know has some noise on it, but hopefully you can see that it is still just cellulose fibers. So because that in situ approach had changed the morphology of how the pellicle grew so much, we actually upscaled it into some materials testing on it, which is what you can see here. So this is tensile strength test. And what we found was that the addition of the hydrophobic protein actually increased the elongation, so increased how much the cellulose stretched before it broke. And particularly for the hydrophobic protein with the cellulose binding module, we found that it also took more force to break it. And from those results, we were able to find out on this modulus, and it had actually decreased when the hydrophobic protein was added. And that just means the material is a bit less brittle. If I bring this back to textiles, if we think about how we use them or how you kind of wrestle into a jumper or your jeans, you need it to have a bit more elongation and be strong. And actually for materials, for engineered living materials, getting the strength to align with a less brittle material is actually quite a challenge. Usually you think we increase the strength, it does tend to become more brittle. So that was a happy extra advantage of using this protein. We could then plot Young's modulus against the density of a bacterial cellulose, and that allowed us to then compare it to other materials. We found that we're staying quite close to elastomers. They're kind of around about here on this graph. And that makes sense if you think about what BsIA is in nature. It creates that elastic skin over the bacterial population. And it seems to be acting in a similar way with the bacterial cellulose. So our conclusions in this experiment were that yes, we can use an engineered biological approach to make bacterial cellulose more hydrophobic. Again, if you remember, I mentioned that we can't exactly say that our orientation is correct. So we might be able to improve how hydrophobic we made our bacterial cellulose. by either looking to alternative hydrophobins. There's numerous other bacterial ones, as well as a whole class of fungal hydrophobins. Or we could go back and modify the protein that we're using at the moment, maybe to try and get that linker region a bit tighter in terms of being able to predict its structure. We also had that increase in other useful properties, the increase in strength, but decrease in brittleness. We think one of the reasons for that might be because the bacterial cellulose had to grow subsurface. Instead of growing as kind of 2D sheets one on top of the other, it was able to grow as more of a 3D lattice. It's just kind of naturally knitted together a little bit more. What would be great to do, and what we'll have to speak to Roberta about, is if we could progress to using it as more of a yarn, because hydrophobicity wise, we weren't getting the full advantage of that in situ treatment, but manufacturing wise, we've functionalized and grown the bacterial cellulose in one step instead of having to do it post-production. And if we could then use it as a yarn, that might mean that we're actually seeing the advantage of the hydrophobicity. This process could be quite a bit more sustainable than what's already being used by industry, but we hope that we could actually push it a little bit further. So we did some quick experiments just to see if that protein was still there. So we degraded the cellulose and found that yes, the protein is still there. So it'd be great to see if we could then reuse that protein for new stocks of bacterial cellulose, as well as reusing the cellulose as a feedstock for a new batch of hydrophobic cellulose. So with that, thank you to everyone for HBBE that was involved in the project, and everyone else at Northumbria. I'll finish there.
Okay, any questions? So have you actually tested it to see if the CDM helps the hydrophobins stay on the surface of the cellulose?
Yes, so if I skip back. So even to this one here, this like cloudiness is all different proteins on the top surface of it. Without the cellulose binding module, it's much less. It is still cellulose there,
More than a little difference in the
which we think could be due to that orientation of what way it's maybe sitting on the surface. If it's not sitting fully outward, we won't get as good hydrophobicity as we could do.
So have you thought about maybe looking at linker sizes and things like that?
That is one thing that we could look at. Yeah, absolutely.
So AlphaFold's notorious for not being able to predict what this is so you might have to go to the X-ray for something.
Yeah, we might have to.
Okay, so I thank you all three of the speakers in this session. Opposed to funny things. So maybe we could give a round of applause to everybody who spoke in this session.